Hello students, welcome back. Everyone likes flowers and use flowers as a medium to express our feelings. Not only that, roses, marigold and jasmine are used for social, cultural and religious ceremonies, especially in India. You might have learnt about floriculture. Floriculture deals with the growing and marketing of flowers and ornamental plants as well as with flower arrangements. But in the view of a biologist, a flower has other functions as well. Flower is the site of sexual reproduction in plants. In this video, let's learn about the structure, morphology and processes of sexual reproduction in angiosperms, that is the flowering plants. Before the formation of a flower, several structural changes happen in a plant that leads to the formation of floral primordium. Floral primordium are small buds from which a flower develops. We know that there are male and female reproductive organs in a flower. Androecium is the male reproductive organ and gynoecium is the female reproductive organ. Let's learn more about the male reproductive structures in a flower. This is the picture of a stamen. Stamen has two parts, anther and a filament. Anther is seen in the terminal end and it has two lobes. Anther is a bilobed structure. Another part of stamen is a long slender stalk called the filament. This filament is connected to the petal of a flower. Flowers of different species differ in their sizes and number of stamens. A typical angiosperm anther is bilobed, that is with two lobes, and each lobe has two theca. The cavity of the anther is called theca. Each lobe has two theca and hence is called dithecous. Dithecous means nothing but having two theca. It is also called microsporangia. Microsporangia is present in young anther. When the anther matures, it is called pollen sac. Here you can see these are the pollen sacs. This is a mature anther, but in a young anther, these pollen sacs are called microsporangia. When the anther matures, it is called pollen sac. There is a longitudinal groove that runs lengthwise separating the theca. An anther has 4 microsporangia. Pollen grains develop in these microsporangia. They are located at the corners. Let's learn more about the structure of microsporangium. When we look at the transverse section of microsporangium, it looks circular in outline. This is a mature anther from which the pollen grains are coming out. But in young anther, the microsporangium is a closed structure. In young anther, this will be closed. 4 microsporangia are present which are surrounded by the wall that has 4 layers. The layers are epidermis, endothecium, middle layers and tapetum. The outer 3 layers, that is the epidermis, endothecium and middle layers, perform the function of protection and help in the dehiscence of anther to release the pollen. The innermost wall layer is called the tapetum. The function of tapetum is to nourish the pollen grains. Cells of tapetum possesses dense cytoplasm and many nuclei. Do you know why? As they help in the growth of pollen grains, they should be metabolically active. This is the reason they possess many nuclei. When the anther is young, a group of compactly arranged sporogenous tissue that occupies the center of microsporangium. As the anther develops, pollen mother cells or microspore mother cells are derived from the sporogenous tissue. This is a microspore mother cell. This microspore mother cell is diploid in nature. That means it has 2 sets of chromosomes. This microspore mother cell undergoes meiosis and produces 4 haploid cells. It undergoes meiosis and produce 4 haploid cells. These haploid cells are arranged in 4 groups and are called tetrads. It will be like this. This is a microspore mother cell, and here these are the microspores. These are the microspore. 4 microspores are produced from a microspore mother cell, and these are called tetrads. A microspore tetrad. This whole process is called microsporogenesis. As the anther matures and dehydrates, the microspore dissociates from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed, and they are released with the dehiscence of anther. Now let's learn more about the pollen grains. Pollen grain is the male gametophyte. If we happen to open the anther of hibiscus, some yellow powdery substance will get deposited on our fingers. These are the pollen grains. If we mix these pollen grains in water and observe under microscope, we can see the beautiful structure of pollen grains. Pollen grains from different species have different shapes, sizes, colors and designs. Pollen grains are generally spherical. The diameter may range between 25 to 50 micrometers. Pollen grain has a prominent 2-layered wall. The outer wall is called the exine and inner wall is called intine. The outer layer, exine, is really hard and made up of the most resistant organic material called sporopollenin. This is the exine layer. This outside layer is the exine, and it is made up of sporopollenin. This sporopollenin can withstand high temperatures and strong acids and alkalis. Sporopollenin cannot be degraded by any of the enzymes. Due to the presence of sporopollenin, pollen grains are well preserved as fossils. This outer layer exine has prominent apertures called germ pores. These are the germ pores. Sporopollenin is absent in germ pores. Do you know why the sporopollenin is absent in germ pores? This is because these germ pores help in the formation of the pollen tube and the release of male gametes during fertilization. As we now know the function of outer wall, the exine, let's learn about the inner wall, the intine. Intine is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain matures, it contains 2 cells, a vegetative cell and a generative cell. This bigger one is the vegetative cell and this smaller is the generative cell. The vegetative cell is bigger and contains irregularly shaped nucleus. It has abundant food reserves. Vegetative cells form the pollen tube that grows to meet the unfertilized ovules. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle in shape with dense cytoplasm and a nucleus. Around 60% of angiosperms, the pollen grains shed at this 2-celled stage, that is the vegetative cell and the generative cell. But in others, the generative cell undergoes mitosis and produces 2 male gametes. Now the pollen is in 3-cell stage, 1 vegetative cell and 2 generative cells. Now the pollen sheds. Once the pollen grains are shed, they have to land on stigma before they lose viability. Then only fertilization will happen. The viability of pollen grains differ in different species. It depends upon the temperature and humidity of a particular area. For example, in cereals such as rice, the viability of pollen grains lasts for only 30 minutes after their release. But in members of Leguminosae and Rosaceae, the viability of pollen grains lasts for months. The pollen grains from different species can be stored in liquid nitrogen for years. These stored pollen grains can be used as pollen banks in crop breeding programs. You might be aware that pollen grains cause allergies, but in some people it can cause serious respiratory disorders like asthma and bronchitis. Now let's see what are the uses of pollen grains. Pollen grains are rich in nutrients. In recent times, people are using pollen tablets as food supplements. In Western countries, pollen products like tablets and syrups are available. Pollen consumption has been claimed to increase the performance of athletes and race horses. As we have completed the structure of androecium, let's move on to gynoecium. Gynoecium is the female reproductive part of a flower. It consists of stigma, style, and ovary. The pollen grains land on stigma. Style is the elongated slender path beneath the stigma, and the basal bulged portion is called the ovary. This is pistil. This whole area is called a pistil. Pistil consists of stigma, style, and ovary. But gynoecium is the collection of all female reproductive system. That means gynoecium may contain a single pistil or many pistils. When there is only one pistil, it is called monocarpellary. And if there are many pistils in a gynoecium, it is called multicarpellary. When there are more than one pistils, they may be fused together and called syncarpous. Inside the ovary, there is an ovarian cavity called the locule. The placenta is located inside the locule. Megasporangia or ovules arise from the placenta. The number of ovules in an ovary may be one, like in the case of wheat and mango, and many in case of papaya and watermelon. Let's learn more about the structure of Megasporangium. The ovule is attached to the placenta by means of a stalk called funicle. Let me draw a funicle here. This is funicle. The body of the ovule fuses with the funicle in a region called hilum. This is hilum. The body of the ovule fuses with the funicle in a region called hilum. That means hilum is the junction between ovule and funicle. The central part of the ovule is called the nucellus. It is a mass of cells. Located in the nucellus is the embryo sac or female gametophyte. Here you can see the megaspore mother cell. This megaspore mother cell which develops into megaspore, which in turn forms the embryo sac. That means embryo sac is located in the nucellus. An ovule generally has a single embryo sac. Each ovule has one or two protective layers called the integuments. Integuments encircles the nucellus except at the tip where there is a small opening called the micropyle. Pollen tube enters the ovules through the micropyle. Opposite to the micropyle is the basal part of the ovule called the chalaza. Nutrients from plants are transported to nucellus through chalaza. The ovules generally differentiate a single megaspore mother cell in micropylar region of the nucellus. That means one of the cells of the micropylar region develops into megaspore mother cell. This megaspore mother cell is diploid in nature. That means it has two sets of chromosomes. It undergoes meiosis and produces 4 haploid cells. it undergoes meiosis and produces 4 haploid megaspores. This formation of megaspore from megaspore mother cell is called megasporogenesis. We now learned that there are 4 megaspores. However, in the majority of flowering plants, only 1 megaspore is functional and the other 3 degenerates. This functional megaspore, this is a functional megaspore that develops into female gametophyte or embryo sac. The method of embryo sac formation from a single megaspore is called monosporic development. Let's learn the formation of embryo sac. The nucleus of the functional megaspore, this is the nucleus of the functional megaspore that divides mitotically into 2 nuclei. These 2 nuclei move to the opposite poles and again undergo mitotic division to form 4 and then 8 nuclei. Among these 8 nuclei, 4 are in one pole and the other 4 in the opposite pole. During these mitotic divisions, nuclear divisions take place but are not followed immediately by cell wall formation. When the embryo sac develops into the 8 nuclear stage, the cell walls are formed around the nuclei. Now 6 of them, 3 are here and 3 are here, 6 of them are covered with cell walls. The 3 cells grouped at the micropylar end constitute the egg apparatus. One of the 3 cells becomes the egg cell and the other 2 becomes the synergids. The egg cell as a female gamete fuses with one sperm cell to give rise to the embryo. The synergid cell walls form a highly thickened structure called the filiform apparatus. Here it forms the filiform apparatus at the micropylar end. These filiform apparatus play an important role in guiding the pollen tubes into the synergids. The synergids brought to an end of pollen tube growth and release of sperm cells. The 3 cells at the chalazal end are called antipodals. The remaining 2 nuclei from the sides move to the central region and fuse with each other. Thus, it is one cell with 2 nuclei. Altogether, a typical angiosperm embryo sac at maturity has 7 cells and 8 nuclei. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.